Yeah.
Okay, no worries. Okay, yeah, so collectively they span over 353,000 base pairs in the bacterial genome, which is almost 7% of the whole genome of this ETEC strain. And now interestingly, when this genome was compared to the genomes of closely related non-pathogenic E. coli strains, such as E. coli K12, it was found that all the coding sequences which are unique to this ETEC strain, 88% are associated with prophages or other mobile genetic elements. Oh, no, it's not. Okay, so given this striking genomic footprint and the lack of knowledge of how prophages contribute to the physiology of ETEC, we aim to explore the functional impact of prophages in ETEC. For that, we generated a mutant library by systemically deleting each of the prophages individually and also all of them in one strain to then in the end have a complete prophage-free strain I'll refer to as delta 8 throughout the talk. And all of those mutant strains, so in total 8 single prophage deletion mutants and the one which lacks all prophages together with the wild type, underwent then subsequently a variety of different phenotypic characterization assays aiming to cover a broad range of traits which are potentially influenced by the prophages. And in the following, I'll present some of the results now. So first, we looked into the contribution of prophages to chemical tolerance or resistance. For that, we used for once so-called phenotype microarrays. Some of you might know what they are. It's basically like 96 well plates, where each well contains a different compound or a same compound in different concentrations to just get a broad overview of which phenotype they might influence or not. So here we use 2 of those phenotype microarrays, which focus on chemicals. So in total, 48 different chemicals in 4 different concentrations were tested for wild type and the strain lacking all prophages. And interestingly, we found that the wild type never outperformed the delta 8 strain across all of those chemical compounds, while delta 8 showed improved metabolic activity for 13 of those compounds listed here in this graph, belonging where the chemicals belong to a variety of different targeting cellular processes. Now, subsequently, we did some minimal inhibitory concentration assays where we used some of the chemicals already used in phenotype microarrays, as well as other antibiotics, which target other cellular processes in bacteria. Here we found a similar trend where the wild type never showed a higher MIC compared to the delta 8 strain, while the other way around, we have found a few antibiotics where the MIC was increased, usually twofold, compared to the wild type. And taking those results together, it suggests that the absence of prophages is beneficial for the bacteria, likely due to the avoidance of stress-induced prophage activation. So next, we looked into acidic stress tolerance, osmotic stress tolerance, and phage susceptibility. And there we found a contrary trend where the deletion of prophages led to a reduced survival in both acidic and osmotic after acidic and osmotic stress exposure. However, for single prophage deletion mutants, we often had only mild effects or sometimes even contrary effects compared to the delta 8 strain. For phage susceptibility, we isolated a virulent phage we named HMGU ETEC1 and from wastewater, and then basically did plug assays with all of the strains to then calculate the efficiency of plating afterwards, and found that delta 8 is more susceptible to this virulent phage we tested, indicated by the higher efficiency of plate or elevated efficiency of plating, as well as constantly larger plaques on the plates. Next, we looked into the different lifestyles of bacteria, basically by looking into biofilm formation and motility, found that Wild type, but not delta 8, was able under our test conditions to form cell aggregates, with aggregation being an early step in biofilm development. And this is then also reflected in the biofilm formation assays, where we've seen that the biofilm formation is significantly reduced in the delta 8 strain, while single prophage deletion mutants rather again showed mild effects or even contrary effects to the delta 8 strain. Then for motility, we tested two different kinds of motility, swimming and swarming. Swimming was only observed for the delta 8 strain, not for any of the single deletion mutants or the wild type, while swarming was observed for all of the strains, however, to a different extent, depending on whether which prophage was deleted. And together, these results sort of indicate that the lack of prophages shifts the bacteria from a more sessile to a more motile lifestyle. Now, to sort of try to see if any of those phenotypic changes then translate actually into changes in virulence, we used an in vivo infection model, namely the Galleria melonella wax moth larvae. And there we injected or infected the larvae, 30 of them each, with the different strains, and tracked activity, melanization, and survival over 5 days. And there we found the trend that the infection with single prophage deletion mutants led to an increased larval activity compared to the wild type, while the delta 8 strain showed that the larval activity, but also survival, when we did Kaplan-Meier survival curves, were lowest when infected with the completely prophage-free strain. Now, after phenotypic characterization, we also just quickly started looking into potential molecular links between those observed phenotypes and molecular mechanisms behind it. For example, did we do some RT-qPCR with small hypothetical prophage genes and in different stress or host-like conditions, and found both prophage and condition-specific expression of those yet hypothetical prophage genes, indicating that they might be involved in adaptive response. We also did some in silico protein-protein interaction analysis, where we've also been able to predict some interactions between prophage proteins and key bacterial proteins which are involved in some of the observed phenotypes, mainly in acid resistance, motility, and colonization. So to sum up, throughout all of those assays we did, we found that complete prophage removal often led to strong phenotypes, while single prophage deletions typically resulted in milder or sometimes even contrary phenotypic changes compared to the delta 8 strain. And this indicates that prophage functions can be redundant, cooperative, or sometimes even antagonistic, with the individual effects often masked or unbalanced in the wild type then. And all of these phenotypes likely arise from a combination of different mechanisms, such as lysogenic conversion, or direct protein-protein interaction, or transcription regulation, or as in case of the chemical tolerance, the lack of prophage induction, for example. And with that, I'm coming to an end and want to thank my supervisors, Professor Li Deng and Dr. Mohammed Ali Khan Mirzai, for their constant support and supervision, as well as everyone involved in this project, and you for your attention. Thank you.